mathematics of molecular motor dynamics, cellular morphogenesis, and tug-of-war. So, thank you.
Okay, thank you. I'm very sorry about the technical problems. So as you can imagine, I wanted to start with this movie up there, which effectively is this one. So it's a kind of a classical movie. It came out 10 years ago, got famous for the rendering actually, and it shows a kinesin motor protein in a cell moving along a microtubule fiber, that is that cylinder here. And and as that motor protein moves with its motor domain, it pulls a fairly huge cargo vesicle through the cytoplasm. So this talk is actually a talk about two different projects, but both projects focus on kinesin motor proteins and microtubules. Again, here we see a schematic view of a microtubule fiber. Microtubules are polar. They have a plus end and a minus end, so the kinesin motor always moves to the plus end. The other motors, for example, those of the dynein family, which typically move towards the minus end of the fiber. There's a standard model for that, and that standard model is what is called this affine force velocity curve. It tells us that if those motors are free to move, then they move at what is called the free moving velocity. And if they experience some force, some opposing force, for example, friction as they move through the cytoplasm, then that slows them down. And if that force they experience corresponds to the so-called stall force, then they get stuck. Then they don't move at all. So then their velocity is zero. A phenomenon which is typical of cargo transport is what is called tug-of-war. So there are situations where there are cargo vesicles that are exposed to different motor proteins which tend to move the vesicle in one dimension, other proteins which tend to move it in the other direction. And typically what happens is that this intermediate situation with equal numbers, or let's say equal forces in one direction and in the other direction, that situation is fairly unstable. What you typically observe is that there are either only motors pulling in one direction or only motors pulling in the other direction. The model that is able to reproduce that behavior, that is this model here for the off rates of those motors. So it's a so-called Bell's law, according to which the off rate increases exponentially with the force that those motors experience. And you can imagine, so if one of those motors, those pulling in a negative direction, if they would attach, they would experience a large force coming from all the other motors. So that one motor would be very likely to detach, while those motors would just keep pulling. I will come back to this in the second part of this talk. In the first part of the talk, I want to present this project here, where we investigate morphogenesis in neuron cells. Neuron cells are the backbone of our nervous system. They have a cytoplasm with a nucleus, of course. What characterizes them is that they have one potentially really long axon, which is this cellular extension here, and many shorter so-called dendrites, which are those extensions here. So those processes, they are used to pass information. Typically, the cell emits information through the axon and receives information through the dendrites. If we look at it more closer, then we see that the mechanical backbone of those processes, that is a bundle of microtubule fibers, here shown within the axon. What you have is, of course, cargo transport along those microtubules to just, for example, to sustain the activity here of the synapse. But growth of the microtubules and also sliding of those microtubules is also responsible for the growth of the axons and dendrites. And that is what I'm interested here. So to start with a few more words about microtubules, as I already said, they are cylindrical. which we see here most notably in the electron micrograph. Often they are aligned radially within the cells with the minus ends at the so-called microtubule organizing center. But in other cells, and especially in interface cells, those microtubules are distributed totally randomly, and that is also the case in the cells that we will be looking at. This here shows micrographs with fluorescent microtubules again, fluorescent in green. So something that really characterizes microtubules, in contrast to actin filaments, is dynamic instability. So I already said microtubules are polars, and at the minus end here they are fairly stable. But at the plus end, those microtubules typically either polymerize, so they elongate, or they shrink. Polymerization is rather steady, while shrinking is pretty fast. And in the electron micrograph, it gives you this characteristic picture here. So it's really a fast decomposition of those single columns here of tubulin proteins. And what is observed is that microtubules undergo alternate periods of growing or shrinking. That is called dynamic instability. So typically they would grow until an event occurs which is called catastrophe. So that is when they start to shrink rapidly until another event occurs which is called rescue. There might be pauses also between growing and shrinking, and then they start to grow again. And so it goes so on and so forth, growing and shrinking. So this just summarizes what I just said. And here we see a light micrograph of an axon where polymerizing plus ends are fluorescent. So this is where fluorescent EB1 protein is bound. Unfortunately, there's a driver problem here. So what I can do is Okay, I just pressed the wrong button. Okay, so this is where we should see something. Yeah, I think I give up on that video. Okay, so what we should see here is an axon, and growing plus ends are fluorescent. And yeah, so these are schematic representations of such a neuron cell. The long extension is the axon. Typically, we observe that in axons, all the microtubules are with their plus end outside. And that implies that they also grow into the direction of the process. So in that movie we just have seen, we should see all those fluorescent points moving towards the tip of the axon, because all the microtubules there are plus and outwards. Things are different in dendrites. In dendrites, we typically find microtubules with both polarities, so either plus and outwards or plus and inwards. And things are also different in nascent axons, so in like young axons. So it's not shown here, but there too you find microtubules typically with both polarities. And yeah, this brings me to the scientific questions I want to address in this project, namely, yeah, on the one hand side, what drives neurite growth? So axons and dendrites both are called neurites. And the other question is, what controls the polarity of the microtubules in those neurites? An experiment people did is this one here. So here we see an axon, and then using genetic tools, kinesin, so this central molecular motor protein was inhibited. And in the absence of kinesin motors, no axons grow. So apparently those kinesin motors, they are the driving force behind growth of axons. And what is quite clear is that, well, we know that kinesin moves towards the plus end of the microtubule fibers. And so even in a situation where kinesin, as it is shown here, would, with its motor domain attached to one microtubule with the cargo domain to the other microtubule, it still, with that motor domain, it would move towards the plus end, and therefore it would push apart the microtubules, but minus end forward. So that could, of course, establish and elongate the axons, but we know that in axons, microtubules are actually plus end forward. And so apparently, it cannot be kinesin alone, because kinesin would push them into the extension minus end forward. So that is a major question I want to ask now. What else is there? What guarantees that actually microtubules in axons are plus end forward and not minus end forward? And so we were collaborating here with an experimental group, and they do experiments not only on neuron cells, but also on those cells here, as two cells. Whenever the actin cortex of those cells is inhibited using drugs, then those cells too, they establish those processes. So pretty much in the same way as axons and dendrites of neuron cells. The advantage of those cells, of course, is that the growth of those processes is really fast. It happens within 1 hour here, while it takes about 10 hours for a neuron cell to grow an axon. The experiments they did were, for example, the following one. So this again shows a control cell. So here we see basically the trajectories of the polymerizing plus ends. And it turns out that in control cells, in those processes, the microtubules too seem to be plus and outwards, because all those trajectories, they are pointing outwards. Here they look at CAMSAP, which is another protein located at the minus end of microtubules, and it's fairly uniformly distributed within the cell, also within the processes. And this here shows the same experiments, but after dynein has been inhibited. So no dynein motor. Now dynein is the motor that moves to the minus end. And suddenly, the trajectories of the polymerizing plus end, they are pointing in both directions here. So here we have bipolar microtubule polarity, like in dendrites. And if you look at the minus ends of the microtubules, it turns out that they are mostly at the tips of the processes. So that is consistent with kinesin alone, just pushing microtubules apart, minus and forward. Yeah, so that is a hypothesis this group formulated in a qualitative way, saying that the processes are initiated through kinesin action, pushing microtubules minus and forward, and that at a later stage, this other motor protein, dynein here, is is coming into play. With the cargo domain, it's attached here to the cortex, with the motor domain to the microtubules. And since dynein now moves to the minus end of the microtubules, it would push any microtubule into the process plus and forward. And if there's a microtubule that is minus and forward, it will push them out of the process. So that is how they believe that things work. Dynein is in those processes and basically does the polarity sorting after the processes have been established through kinesin. And yeah, this time we were asked to come up with a mathematical model in simulations to verify that quantitatively. And let me just briefly go over the mathematical model. So it's basically a Brownian dynamics simulations where we look at microtubules, and they are represented as a chain of nodes and springs, as it is shown here. What is also simulated is the action of kinesin motors here at the intersection of microtubules, of dynein motors shown here. They are located at the cortex. Yeah, let me just briefly go over the forces that are taken into account. So as always in cell biology, things are set in the overdamped regime, so I neglect inertial forces. I take into account friction with the cytoplasm. Then this is to guarantee that the distance between the nodes corresponds to a prescribed value here. This corresponds to forces due to bending elasticity of the microtubules. We have thermal forces. We have repulsion at the cortex, which is, as far as the force balance equations are concerned, which is basically a given boundary to the interior of the cell. And these are the forces due to dynein motors and kinesin motors. And as I said, so the kinesin motors, they are active here at the intersection of microtubules. And since the microtubules are a chain of segments, actually those intersections correspond to an intersection of one segment in one microtubule and another segment in another microtubule. And what I have to do is I have to basically compute those forces here and redistribute them to those nodes. And this, well, same thing for kinesin and here also for dynein. And yeah, these are the forces. So I only will go briefly through it. So most of those forces are formulated in a variational sense as a gradient of an energy function like this one here. So this guarantees equal distance between the nodes, for example. Yeah, this is the repulsive potential of the cortex, for example. This is the bending energy, just the deviation of the directions of neighboring segments squared. And yeah, what is interesting actually is how dynein motors and kinesin motors are modeled. So I said that the standard model is this affine force-velocity relation. So basically a constant plus something linear. If you translate it into energies, it becomes something linear plus something squared. The v here is a discrete velocity. And yeah, with that model, so that is the Brownian dynamics part of the model. The model is somehow bigger. So what I actually do is I go through all those 4 steps in every time step, and those 4 steps are, well, turnover of the motors, because the motors diffuse, of course, they detach, they attach. Then I solve this system of force balance equations. Once this is done, I also look at the position of the cortex. So as far as the force balance equations are concerned, the cortex is like a fixed wall. But then I evaluate the forces with which those fibers push against the tip of the processes, and if those forces exceed a threshold value, I elongate the process, so from here to there, for example. And the last step is to simulate also microtubule dynamic instability, so microtubules are growing or shrinking. And if I do go through all these steps, I get simulations like this one here. So this is on a fairly fast time scale. So you don't see very well what is going on inside of the cell, but yeah, it shows how the processes grow. And I track, of course, the length of those processes as a function of time. Now this goes so on, of course. This shows, I think, even the very same simulation, but on a much slower time scale. So what you see here, so that the thick, short bars here, these are the motors. So the blue ones are the kinesin motors at intersections of microtubules. The red bars here are dynein motors. They're always located at the cortex. And the microtubules, they have different colors. So they are dark blue if they are currently being sled by a kinesin motor. They are red if they are currently sled by a dynein motor like this one here. And otherwise, they are light blue. Here, for example, you see how this minus ends, so the minus ends of the fibers are represented as small circles, how this minus end pushes against the tip of the process and pushes it outside. And sometimes you also see a plus end. Well, not now this plus end just reached the tip of that process. And here, this plus end also tries to reach the tip of the process. Of course, it can't, and you will see in a minute why it can't. Yeah, so the reason why it can't is that dynein has a significant effect whenever dynein is in those processes, like those here. But typically, dynein motors are weaker than kinesin motors by a factor 3 or 4. And so you need those processes to be fairly long, as that one here, such that there are enough dynein motors in the process attached to a given microtubule, such that they can overcome the force that might be exerted by a kinesin motor here in the cytoplasm. Yeah, so you need to just wait until the processes grow long enough such that dynein can have an effect. And so, yeah, the picture that we got is the following. As it was hypothesized by the biologists, process initiation is through kinesin in the cytoplasm. You can see it here. So typically, the way processes are established is through those minus ends here and there. Sometimes you also get a plus end, like this one here, but this is rather rare. And then later on, if the processes are long, such as this one here, they are suddenly filled with plus ends that polymerize into the process and keep being pushed towards the tip of the process by dynein. On the other hand, in dynein knockdown cells, so there's no dynein in these cells, if you look here at the tips of long processes, then again, you will find them only minus ends. So in this case, kinesin, which is in the cytoplasm, can push the microtubules even into the long processes minus end forward. And yeah, so what we did is, so that group of biologists, they did a few experiments, for example, those here. And this is now in order to look at how fast the processes grow. They just took those S2 cells, plated them in a petri dish, and took videos for about 1.5 hours. And then after that, what we did is we tracked single processes as they grow. So here, the process is just started to grow. So here, apparently, it has 2 millimeters, then it keeps on growing 5 millimeters, 10 millimeters, and so on and so forth. And here I plotted those lengths of various processes against time. This is now for control. This is for dynein-inhibited cells. And this is the respective simulation result. So yeah, at least something similar is going on. So in control cells, we have those processes grow, but later on their growth is just much slower. A similar thing happens in the simulation. So apparently here there's the threshold length where dynein is active. And in D minus cells, that growth is sustained. Yeah, just apparently the processes can grow longer, and the same thing happens also in the simulations. And this here, well, this upper row here shows the same data, but now visualized with box plots. So what we observed in the simulations is that especially for dynein to be active, we need dynein to be in contact with plus ends. So here, for example, in control cell, the idea is that kinesin drives process growth up to a certain threshold length, and then it's dynein which takes over. Of course, dynein to take over, it needs plus ends of microtubules, but we argued that if there wouldn't be any dynamic instability, so no microtubule polymerization, then those plus ends would never reach the process, and so dynein wouldn't be active. Based on that hypothesis, we asked the biologists to stabilize microtubules with a drug called Taxol, for example, that can be done. And so they redid the same experiments, now applying Taxol to those cells. And this is the data they got. So in both cases, control and dynein knockdown, process growth was largely inhibited. They still did grow, but much slower and to shorter lengths, as it is shown here and there. And in simulations, we got the same result. So apparently, really, in the absence of dynamic instability, dynein certainly cannot be active, so the processes stay short. And also in the absence of dynein, the processes are shorter, and apparently the reason is that as kinesin grows the processes, it needs the microtubules to grow. I mean, the longer the microtubules grow, the further kinesin, which is in the cytoplasm, can reach into the processes and keep pushing them outwards. Of course, so without dynamic instability, the microtubules stay fairly short, and that limits the effect kinesin can have. So that explains why also in dynein knockdown cells, process growth is attenuated as compared to cells without any Taxol. Okay, so this somehow confirmed that the model works, and the model, so this now summarizes the effect of Taxol. As I just said, in both in control and dynein knockdown, process elongation is slower if microtubules are stabilized, and this is consistent with simulation results. And I already gave you the explanation for that. So in control cells, the reason is that dynein cannot be active in the absence of plus ends, and you need. And so if microtubules do not grow into the process, dynein can't have an effect. This is what is written here. And in dynein knockdown cells, it's basically because the microtubules stay short. Okay, and let me just summarize what we learn now from this simulation project. The most important message is that dynamic instability has a major effect. So this is something that even the biologists were not aware of. And other things we learned were that actually microtubules in those cells grow fairly long. It wouldn't work if the microtubules stay short. And we also learned that dynein actually does not do polarity sorting. It is rather a gatekeeper that doesn't allow minus ends of microtubules to enter long processes. Okay, so these are the lessons learned from this modeling project. And now I would like to talk a little bit about another project which deals with tug-of-war between antagonistic motors. We already went through this sketch here. And what can explain this behavior is this force-dependent off rate. So a situation where there's pretty much an equal number of motors pulling in one direction and an equal number of motors pulling in the other direction, that situation is unstable. You would rather have all the motors pulling in one direction or all the motors pulling in the other direction. And these are the trajectories of cargo vesicles, which are typically observed. So time and distance. Typically, we get fairly long run lengths and run times, and then there might be a pause, and then the cargo might switch direction because the dominant group of motors might have switched. What we are interested here in this project is to compute the frequency with which that happens. And to do that, we came up with a fairly simple basic model. And we used this specific model system, which is even easier simpler than vesicle transport. So here we are looking at pairs of microtubules instead than at looking at vesicles. So two parallel microtubules, let's say both is a plus and to the right, and they might interact, might be attached to kinesin motors, like those here, shown in green. And if all the motors of all the if all the motor domains of all kinesin motors were attached to one of the two microtubules, let's say the top one here, they would, well, the motor domains would move to the plus end. That would push this microtubule to the left in minus direction, and this microtubule here to the right in plus direction. You might also have the opposite situation, where all the motor domains of the motor proteins are attached to the bottom microtubule here. And still, so those motor domains would move to this plus end, which would push that microtubule here to the left now, before it was to the right, and that microtubule here to the right, before it was to the left. So we also have a system of antagonistic motors, but it's actually the same motors with the very same characteristics. The only difference is that in one case they are aligned like that, in the other case they are aligned like this. And of course, you might have this intermediate situation where there's an equal number of motors aligned in one way or the other way. And in that case, we would expect that the forces exerted by those motors would balance each another, and those two microtubules would be locked. Now, this is the model we formulated. So first, a very general approach where we say that, yeah, we formulate a continuous time Markov process here. And the state space is the number of motors being attached. So n, it's this tuple here. N is the number of motors in plus direction. M is the number of motors in minus direction. Capital K is the bound of the total number, on the total number of motors. And so the state space is, well, these are those points here. And we have transition rates. So if you're here, for example, 3 motors to the right, 2 motors to the left, then we might just add some motors, either to the right or to the left, at the rate beta, that's a given constant rate. Or those motors might detach, and that happens at those rates here, kappa minus, kappa plus. The difference is that, well, the on rates are assumed to be constant, the off rates are given by those exponentials. And, well, those off rates grow exponentially with respect to the force to which the motors are exhibited. So we need to compute those forces, and that means we need to come up with some model for the mechanics. And we just took the simplest possible one, assuming that there is a force balance now between motors pulling to the right and motors pulling to the left. And every single motor exerts a force according to this affine force-velocity relation, so a constant minus plus a velocity. And, well, that allows to compute those forces here, given by those expressions. So, for example, the force to which a motor pushing to the right is exposed is given by the number of motors to the left times this factor here, 2 over, well, total number of motors. That still turned out to be fairly, well, that seems to be a rather realistic model, but still rather complicated in order to compute this when it comes to computing the switching frequency. So what we did is we simplified that even more, and we were assuming that the total number of motors is given by a constant now. And yeah, that is the idea. So you have this pair of microtubules with motors aligned in one of the 2 possible directions. And what might happen is that one of those motors, for example, this one here, is replaced by another motor aligned in the opposite direction. So we are assuming that basically the off rate is the bottleneck and the on rate is very, very fast. So this happens, so replacement happens immediately. And so as far as the state space is concerned, we stay on this linear chain of states here. So either all motors to the right, 0 to the left, 1 to the left, all the others to the right, 2 to the left, all the others to the right, and so on and so forth. The rates are given by those expressions here, so it's still exponentials, where the number of motors, well, I is the number of motors to the right now, and in those rates you have this I popping up twice as a factor in front and in the exponential. The gamma is the scaled stall force, And basically the idea is that the larger this gamma, the larger the effect of tug-of-war. So yeah, this is just a generator matrix for this process. And one straightforward thing you can do is you can do stochastic simulations like this one here. And you see, well, the effect of the exponential off rate. So this is now, this shows the tug-of-war. For the first like 300 units of time, all the motors are pulling to the right. Then by this rare event, the system switches towards the left. So now almost all the motors are pulling to the left. Then it switches back to the right, at some point back to the left, and so on and so forth. And yeah, as I already said, what we want to compute is the frequency at which this happens. So another straightforward thing you can do with this process is formulate the forward equations for the probability to be in a certain state I. And you can also compute the stationary distribution of this process, which gives you a distribution like this one here. And it also shows you that most of the time you either have all the motors pulling to the left or all the motors pulling to the right, and only very rarely you are somewhere in the middle. Of course, this still doesn't answer our question. How often does the system switch from left to right or from right to left? Yeah, so I would say there are two approaches to answer that question. One approach would be to look at the spectral properties of this matrix here. So there are theories. So for example, a thing called eigentime identity, and there's also a theory of metastability, which tells us that it's basically the dominant eigenvalue here, the slowest eigenvalue after eigenvalue 0, that corresponds to this frequency. But I mean, that wouldn't allow us to compute an explicit formula, which is our goal. So we choose another approach, namely we were looking at expected hitting times. So this is the expected time to go from any state I to any state j. How to do that? That is fairly standard. So if you would look, for example, at times to go from any state I to state, let's say 5, you basically would take the process and make state 5 absorbing, which basically means that you skip column number 5, or in this case 6, and row number, in this case 6. And you get this modified matrix here. And the linear system that based on this matrix and right-hand side ones, that gives you the expected hitting time. So that's a standard result. It can be made more intuitive if you just imagine that according to this system, the expected time to go from state I, let's say, to j is given by the expected time to go away from I, plus either the time to go from I plus 1 to j or I minus 1 to j, and weighted with the probability that you actually went to I plus 1 or that you actually went to I minus 1. So that is actually just another way to write this system here. And yeah, in the case of birth-death processes, so these are the processes where all your states are aligned linearly along a chain and you only have transitions between neighboring states. In the case of those special processes, those expected hitting times can be are given by a fairly simple expression, namely this one here. It's just a sum, a double sum of those products of rates. So one way to understand that is just to solve this system here, like brute force. So these are the determinants which are involved in computing this solution. These matrices can be simplified. And in the end, you realize that things cancel and you get this expression. Another way to see it is to solve this system here. So that matrix here times this vector to write down an iterative solution. So yes, how do you say a recession? Okay. So in any case, I mean, there are. You mean this one for the expected hitting times? No. This you solve numerically.
That's a recursive equation, right? That you solve it and you found CPI.
Okay, that's the word I was looking for. So you write down a recursive solution for this system here. That one times this vector equals 1. That works. But for this, you would compute a numerical solution. Okay, so this here basically gives us a first result. We could, for example, look at the expected time to go from state 0, so all the motors to the right, to state J, and J would be pretty large. So just to make sure that we go to the other side. And we already would have some estimate for this time until the switching occurs. Of course, those kappas here in our specific case are given by specific expressions, namely those exponentials. Those here. And at this point, of course, we also realize that this is quite convenient because here we have products of exponentials, and they can be written as sums, as exponentials of sums, as I did here. And still, the project is about computing a closed form expression for this frequency. And so at this point, we started to simplify this expression as much as we could and to look at asymptotic solutions for this expression. One thing you can do is you can write this double sum here as a double integral, which is an approximation for large K, so large total number of motors. And then you realize that in the integrand, you get this expression here, which is, and the numerator and the denominator actually have the same structure. Both are given by this function f here, evaluated at one variable integration or at the other variable of integration. And if you look more carefully at this integrand, you also see that it has one pronounced peak. In this case, it's that one here. And so this suggests to use what is called Laplace method to approximate this integral, where you basically replace that integrand here by a parabola, which approximates the integrand right at the maximum. So this is what I wrote out here. Of course, so what we do is basically we extend the domain of integration from this triangle to the entire R2. And of course, the peak of this integrand is where the numerator is maximal and the denominator is minimal. So basically, as far as this function f is concerned, we look at the maxima of this function f and the minima somewhere here or there, symmetric. And so what I do here is I take this integrand and I replace it by a Taylor approximation of this function f at its maximum and in the denominator at its minimum. Of course, this I can solve explicitly. It gives me that expression. And the maximum of this function f is always at 1/2, so that is straightforward. The minimum, that cannot be computed explicitly, but it can be approximated for large gamma. Gives you that expression here. And so if I plug in everything, I get this closed form approximation here for the switching time. In this case, it's the expected hitting time to go from one extreme to the other extreme K. And so this was quite a nice result that we were able to estimate this switching time, and hopefully it's useful to experimentalists and can be tested.
So what's the difference between X capital M and X small m?
So X capital M is my notation for the position of the maximum of this function, which is always 1/2, and X sub lower capital M is my notation for the position of the minimum, so either here, there, or here. Okay, now let me come back to the stochastic simulation with which I started. Here we can measure that the average period between switching is 162 seconds. And if I now evaluate those formulas, then I get this result. So this is a log plot. This is the logarithm of this period. And for the first formula we had, so this is just the expected hitting time in the discrete case that corresponds now to this light brown curve. So that seems to be pretty exact. So really for gamma equals 3, which is the parameter I used here, that number here, 5 point something, is really the logarithm of 162. Yeah, so what is not correct here is actually the increase of this period for low gamma, because what you would expect is that the lower gamma, the faster the switching. So the increase here is an artifact, actually. The reason is that here I'm plotting the switching time from 0 to k, the other extreme. But if gamma is small, then actually most of the time we're pretty much in the middle. We only rarely reach the extreme case where all the motors are pulling in one direction. So this increase, yeah, that explains why we have this increase here, and it can be removed easily if I just do not go from 0 to k, but from 0 to, let's say, the maximum of the stationary distribution on the other side. And the other curves here are the approximations. So this is the approximation with the integrals, which is also pretty correct. This is the approximation for large K and large gamma, which gives me the closed form expression. And yes, it's only fairly correct for large gamma, but that was to be expected. Nevertheless, we learned that those switching, so the period of switching is an exponential, basically, of both the total number of motors, K, and the scaled stall force gamma. Okay. Yeah, at this point, let me just summary again. So yeah, as I said, there are actually two approaches to look at those switching times. We chose the one based on expected hitting times, and that allowed us even to compute a closed form approximation to this switching frequency or switching period. And yeah, what we will do in the future is, of course, compare with experiments and also look at the more general model where the total number of motors is not fixed to be some constant, but maybe bounded. Yeah. At this point, I thank you for attention and ready to take your questions.
Thank you. Are there any questions?